and different platforms. So now let's see the principle behind NGS, like how it works. So exactly. So the now I'm sure you all understand like NGS is to know the sequence composition of our DNA or our genetic material in our cells. So how exactly are we going to do it? Right, so the sample, we can extract the genetic material from variety of samples. So for example, the sample could be a blood sample, or a tissue sample, or a saliva sample if we are trying to understand the genetic material of human samples. And if we want to understand the genetic material of microbes present in our environment, so for example, the microbes in the soil, so the soil could be a sample. And like that, like there could be many types of samples from which we can extract the genetic material. So nucleic the first step is extracting the genetic material, let it be DNA or RNA. So then the quality check on the wet lab side. So then the second major step of NGS workflow is library preparation. So when we extract the DNA or the genetic material, we have like amount of DNA present in our hands. So then the technique works like fragmenting the entire DNA using certain techniques on the wet lab side, and we add adapters and indexes according to the problem we are trying to solve, and we amplify it for improving our confidence in sequencing. So once that step is done, the third step is sequencing. So after all the DNA or the genetic material is processed, we load the sample onto our flow cell, or that's a equipment on the sequencing machine. So we load the samples onto that, and that flow cell is inserted into the sequencing machine in order to get the information of what bases are present in the DNA sample we are analyzing. So all the sequences or all the bases are read based on fluorescence technique, and specifically on Illumina, it is sequencing by synthesis. So when each base gets added to the flow cell or the primers, so then it's get recorded, and that is how we get the raw data files. So the raw data files contain all the information of what the composition of our DNA fragments present in our sample. So all that is given as a raw sequencing file. Then the last step is a data analysis. So now we have the raw data files. So as a computational biologist, like our role starts here, begins here, where we start working with the raw data files, quickly do the quality check and aligning it with the reference genome to check where the genetic changes are present in the sample of our interest, and the further downstream analysis. So major